Okay, I think we'd like to go ahead and get started with that presentation now. So without further ado, let me introduce David Butler. He's the Chief Technology Officer at Hongene Biotech Corporation. And today he'll be talking about chemoenzymatic ligation for siRNA and sgRNA manufacturing, recent advances and development strategies. Over to you.
Thank you very much, and welcome everybody to the presentation. So let's dig straight in. Disclosures: I'm an employee of Hongene Biotech Corporation, and I serve on the board of directors of Acta Therapeutics. For an overview, I'll give a recap of some stuff that was discussed last October at the OTS conference, because it's important for understanding this presentation. And then I'll talk about chemoenzymatic ligation for siRNA synthesis with a couple of case studies. I'll talk about chemoenzymatic ligation for sgRNA synthesis with a case study, and I'll also talk about our partnership with Resi Biopharm. And then I'll give a taster of some data that we've generated with our thermostable engineered T4 RNA ligase. I'll also talk about work that we're doing on control of stereochemistry with Bruker, and some conclusions to finish. So there's plenty of information in this OTS presentation, so I put my email address here. If anybody wants a copy of that deck, they can reach out to me on my email address. If anybody has incurable insomnia like I do, then you can also watch the presentation again on Vimeo, and that's linked right there. But to recap on the salient points from that presentation, so there's a huge market potential for siRNA drugs. It comes from the potential demand for drugs like the ones in this table, the 6 drugs in this table, all targeting cardiovascular disease in one way or another. And by our reckoning, this could create a huge market demand by the early 2030s of maybe tens of tons of API for siRNA. And of course, that is going to put enormous strain on the current methods of manufacturing. And on the right-hand side, we all anticipate, hope, and expect that the siRNA therapeutics and oligotherapeutics in general are going to mirror what we've seen for biologics and the life cycle there. So at Hongene, we think about oligosynthesis technology in terms of 3 generations. And on the left-hand side, this is generation 1 technology. It's the phosphoramidite method. And it's been about 40 years in development, and we're standing on the shoulders of giants as far as this technology is concerned. And it's really served us excellent over the years, and it's got us to where we are. But we're seeing some change coming. So on the far right-hand side, we have generation 3, and that's processes that mirror biological processes. And the people here are making fantastic progress. So look at the work from Codexis and N+1, for example. And there's no reason, really, that this technology can't be the endpoint for the industry with the right engineering of enzymes and processes. But slap bang in the middle of these is generation 2 technology, which is chemoenzymatic ligation of oligonucleotide fragments, and that's the focus of the presentation today. We believe that this is going to play a huge part in oligonucleotide manufacturing over at least the next 10 years. And the advantages are clear for us: better yield, better purity, environmental sustainability, and scalability. So what's Hongene's process? Well, we have 2 processes that we use, and the first one is sticky end ligation for synthesizing siRNA and similar molecules. And this technology has really been around for a very long time. Over 50 years, Gobin Khorana first described a similar approach. And this has been exemplified specifically for siRNA back in 2014 by Sosik et al. So we use essentially the same process. So we take 4 oligonucleotide fragments and we hybridize those in the first step to form 2 duplexes that have these sticky overhangs that then cohere, and the junctions that form between the fragments are ligated with ligase. The siRNA duplex is formed directly, and that is purified by non-denaturing column chromatography. On the right-hand side, meanwhile, we have our process for making single guide RNA and long RNAs in general. So we use splinted or template ligation for this process, where we take a certain number of oligonucleotide fragments, depending on the length of the molecule, with templates, reverse complementary templates, which are DNA, and then we hybridize them. We ligate the junctions between the fragments, and then we digest the DNA templates with DNA SWAN. And that allows us to get the single guide RNA, which is then purified by column chromatography. Okay, so let's jump into some of the work that we've done recently with chemoenzymatic ligation for siRNA synthesis with a couple of case studies. And the first case study we're going to look at is inclisiran. And so inclisiran, I think, as a molecule, it needs no introduction to this audience. Suffice to say that it contains the chemistry that's typically found in the siRNA therapeutics that were in that table. So you've got 2'-fluoro, 2'-O-methyl. There is a deoxy in the inclisiran molecule, which is a little bit unusual, but otherwise it's the same kind of chemistry. The backbone's PO with PS's on the ends and a triantennary GalNAc. So it's really very familiar, and it's an excellent model. It's commercially validated, so that's why we selected it for our proof of concept studies. And so how does our process look? Well, first of all, we have the oligonucleotide fragments, and these are synthesized using generation 1 chemistry, right? So solid phase chemistry. Then we do the C&D. We purify the fragments by column chromatography, and then we desalt and we lyophilize. The lyophilization step is completely optional, and when we scale up, we actually omit the lyophilization step. Because this is proof of concept, small scale, it's just convenient for us to lyophilize the material. So those oligonucleotide fragments are then fed into the chemoenzymatic ligation reactions. There's hybridization, ligation, and then we do a purification, that's non-denaturing chromatography, desalt, and then lyophilize to get the product. And we term this process P2P, right? So this is the P2P process. It means we're purifying the fragments and we're purifying the siRNA. And we control the quality of the fragments with IPR, PHPL, CMS. And as you might expect, because these have been purified by column chromatography, the purities are all really high, right? They're all above 95% purity. The yields, I would say, are moderate, right? We're looking between 35 and 55. But these are completely unoptimized. So there's plenty of room for us to optimize the yields for these fragments. And then we mix these together, and we subject them to the chemoenzymatic ligation, and we measure the crude HPLC, which you see here. It's the blue trace, and that's 95% pure. Meanwhile, the black trace is the purified material after downstream processing. And the first thing that jumps out is that you have very high fragment conversion, and in fact, the conversion, it's a quantitative conversion in the limiting strand. So if you get the stoichiometry of these fragments just right, then you're not going to see very much in the way of fragments in the crude material at the end, and that's what you see here. Digging down a bit on the impurities, you can see that these impurities in the table that are in blue, these are impurities that are in the crude, but they're not in the purified material. So these are purged, and these are fragments and fragment-related impurities. Meanwhile, the impurities that are in black, these are solid phase, typical solid phase-related impurities. So importantly, there are no new ligation-related impurities. And these solid phase-related impurities that you see, these are typical of what you might expect, right? There's PS to PO, there's N+1, N-1, M-2 and M+12, these are actually GalNAc-related impurities. We see also M-20 and M-2, and I'll talk a little bit about that in a minute. But any one of these impurities is present at less than the 1% ID threshold that was suggested by Capaldi et al. In 2017. So on the -20 and -2, this comes up in conversation, right? There's still people that don't know completely about the origin of these impurities. So this is just to put that out there. So -20, that comes from the intramolecular degradation under the action of base and heat. of the 2' fluoropyrimidine to generate this 2' anhydropyrimidine. So that's minus 20. And then that undergoes base catalyzed hydrolysis to generate this arabinosyl pyrimidine. And so that's well described in the literature. And this is aligned with our observations. So we see elevation of these impurities following lyophilization, in fact. But we also see a change in the distribution of these impurities on storage, either in solid or in solution. So taking a step upstream, the oligonucleotide fragments are a really important focus of impurity control for us. And really, these are the reasons that this technology, we're able to get improved purity for generation 2 versus generation 1. And broadly speaking, there are 2 reasons for this. So firstly, all those solid phase related impurities that we talked about, those are generally greatly reduced in the fragments compared to what you would see in a typical full-length sequence manufactured with generation 1 technology. And we think the reason for this is quite simple. There's actually just less opportunity for these types of impurities to compound and accumulate for a shorter oligonucleotide compared to a longer oligonucleotide. Secondly, some of the oligonucleotide fragment impurities, they're just not great substrates for the ligase. So the ligase is good at deselecting for oligonucleotide impurities. So for example, in the fragment 4 on the right-hand side, these impurities that are in red, these are impurities that lack the 5' phosphate on this fragment. And so these can't possibly ligate. They're not a substrate for the ligase. And so these are removed during downstream processing, during purification of the siRNA. Meanwhile, the impurities that are in black in this table, these persist. And that's as you might expect, right? A PS to PO, that is clearly going to hybridize well and persist into the product, and also the -2 impurities here. Another case study is this divalent XNA containing siRNA. And this originates at UMass. So they invented this technology. The XNA contains this additional carbon atom on the 5' position of the nucleoside that imparts additional stability against nucleases. And then you have better duration for the drug, better efficacy. And so that's the XNA technology. So scientists at Broad, UMass, and Harvard recently identified a clinical candidate based on this chemistry for prion disease. And Hongene was very fortunate and honored to be selected as a CDMO to manufacture this clinical candidate. And we did this using exclusively solid phase synthesis, so generation 1 technology. But of course, we were naturally curious about this molecule and whether we could apply the chemoenzymatic ligation technology about it, because it's a nice stress test for the technology. It's new chemistry. It's a divalent siRNA. So looking at the chemistry below, we chose a model for this, and this model chemistry has the XNA modifications on the 3' end and a vinyl phosphonate on the 5' end of the antisense strand. Meanwhile, the 2 sense strands are linked with this tetraethylene glycol linker. And this is the chemoenzymatic process design. So it's almost the same as we described for inclisiran. We're using the P2P process, and you might think there'd be a larger number of fragments, right? But by virtue of the fact that it's a symmetrical molecule, you still only have 4 fragments. And we control the purity of those with our IPRP HPLC-MS method, and the purity is, as you would expect for purified fragments, always greater. It's greater than 93%. Again, the yields are kind of moderate to good, but we haven't optimized those at all. So those fragments are then fed into the chemoenzymatic ligation reaction, and this is what we see. So the trace in blue is the crude mixture, and the trace in black is the purified siRNA. And the first thing that jumps out, again, very high fragment conversion, very high purity. Almost 97%. And having made a similar molecule with the solid phase chemistry, generation 1, I can tell you that this purity compares very favorably with that. And again, quantitative conversion in the limiting strand. And taking a deeper dive on the impurities again, the impurities in blue, that's A through E. These are all fragment-related impurities, and these are easily purified by downstream processing. So there are no new ligation-related impurities in this purified material. Also, the impurities that do persist in black, these are similar to the impurities that we saw before. There is one new one that we're still noodling on. That's the one that's plus 126 Daltons. So we're still trying to identify that one. But it's present at a very low amount, and all of the impurities that persist are below the 1% ID threshold that's suggested by Capaldi et al. So to summarize on the siRNA piece, we have inclisiran and the divalent siRNA case studies as the top two lines in this table. These are the P2P strategy. I'm not showing the data for the C2P, but we have a C2P example in this table. It's the middle line. So when we go to a C2P strategy, that's crude fragments, and you're purifying the siRNA. You see a really significant improvement in the yield. Again, these are not optimized yields. So for P2P, we're typically seeing 20 to 25%. For C2P, we typically see 40 to 45%. But you can see you're not really sacrificing that much in terms of the purity. It's still better than it would be for a generation 1 synthesis for sure. So there's big advantages to removing the column chromatography for the oligonucleotide fragments in terms of cost. The bottom two lines in this table, this is the same siRNA that's been into our manufacturing CDMO services, and the second bottom line is the engineering batch that was synthesized using the C2P process, and you can see the purity was 95%. Meanwhile, the bottom line is that same siRNA that has been through our GMP facility. We manufactured 1 kilo of that material, but we used the more conservative approach, right? We used P2P strategy for that. But importantly, we've synthesized this now into GMP. It's been past regulators, and this material is being dosed in patients. So this technology is here and now. The C2P is working great. C2C, we're curious, right? Well, can we use C2C? So that would remove column chromatography entirely from the fragments and the siRNA, so you'd be processing by UFDF or something similar. And this could provide the advantages that you need in terms of the cost for these large volume cardiovascular disease drugs, whilst hopefully providing the necessary quality characteristics. The chemoenzymatic ligation is adaptable to batch mode synthesizers and single-use bioreactors. The examples that I showed you were about 20 grams per liter, that type of concentration, but with other process development that we've performed, we've gone to at least 100 grams of siRNA per liter, and that really helps with the scalability of the process. We see no reason why this can't be a process where you can manufacture hundreds of kilos of siRNA. And of course, there's plenty of room to optimize the oligonucleotide fragment processes, and that's not something we're going to dig into too deeply today, but there's plenty of people working on getting the solid phase out of the flow-through reactors and into batch reactors. People are talking about that at this conference, and also solution phase synthesis. So these will become much more important technologies as the requirements for these volumes increase. Next, I'll talk about chemoenzymatic ligation for sgRNA synthesis. And the case study here is G211 sgRNA. And we selected this sgRNA. This comes from an Intellia paper where they were looking at in vivo gene silencing of TTR. And it's a great model for us. It has a highly modified sequence. There's a lot of 2' methyls in it. There's 2' OH and phosphorothioates on the ends of the molecule. And this is typical of the type of chemistry that you might find in other clinical candidates. So it's really relevant and an excellent model for us. So we employed a 2-step splinted ligation strategy, and the purpose here was to get ultra-pure, ultra-high purity guide. So the guide sequence is this 20-mer, and that does the targeting, right? So we wanted to get the highest fidelity possible. So we actually synthesized that also using splinted ligation in step 1 from 2 shorter fragments, and we obtained material that had that quality, that had that excellent sequence fidelity. And then in step 2, that G21120mer is assembled with CR1 and CR2. Those are the other 2 oligonucleotide fragment components. Those are 40mers, and they're assembled by splinted ligation with DNA splint 1 and 2. The process flow is very similar to what you saw for siRNA. So the DNA splints, these are used crude. There's no column chromatography for the DNA splints. Meanwhile, the oligonucleotide fragments, we are purifying these by column chromatography. And for the chemoenzymatic ligation, there's an additional step upstream. So we do the hybridization ligation, but then we have a digestion of the DNA template by DNase 1. And then we have the purification by column chromatography, desalting, and lyo step. And these are the components that are fed into the chemoenzymatic ligation reactions, these step 1 and step 2. So you can see that the G21120mer coming out of that step 1 synthesis has exceptional purity, right? It's 98% pure. CR1 and CR2, meanwhile, are less pure, but they're satisfactory for this reaction. And the DNA splint 1 and 2 are crude, so there's a set between 70 and 80% purity. So these are all the components that went into the reaction. And when we do the chemoenzymatic ligation, what we're looking at here is the blue trace is the crude reaction before, so this is after ligation, but before digestion of the DNA splints. And the black trace is the purified single guide RNA following downstream processing. So again, the first thing that jumps out to you here is the exceptional purity value, right? The HPLC purity is above 95% pure. And this compares with what you would see for a generation 1 synthesis, where you're struggling to get, sometimes 75 to 85% purity with an HPLC method that is good at resolving impurities. And we have partners that we're working with that are helping us to validate this purity. And so far, this purity value is ringing true. We're getting that kind of feedback from our partners. Again, if we take a deeper dive on the impurities, we can see that all of the impurities in blue, these go away with DNase 1 treatment and purification downstream processing. So these are fragment-related impurities and things that are ligated, but maybe not all 3 of the fragments are ligated. Only 2 of them are, like E and G. These are all easily removed by column chromatography. There's an impurity here that persists. Again, it's that M+126. So if anybody has any ideas what that might be, please let me know. But that persists into the product. So we're trying to work out to figure what that is. That's present at 1.6%. We're working hard to build our capabilities around HPLC and mass spec. As you can imagine, there's a heavy reliance on these technologies for the analysis and control of these compounds. So this is some examples of the work that we're doing. So we're looking at this as a 99mer. So this is 5' N-1, and we're spiking it into the purified material. And then we're taking extracted ion chromatograms across the region where the 5' N-1 is being partially resolved, and we can identify that by mass spec as well. So we're building these capabilities. We'll do more work here to look at other impurities. And for mass spec, we understand that we need intact mass identification. It would be great if we could get impurity profiling with a single Dalton resolution. So you could tell C from U and A from I. And also, we're developing methods for sequence confirmation. So we're really building these capabilities in this area. A little bit on our partnership with Resi Biopharm. So the value proposition here is that you get gene editing, drug manufacturing under one roof in Massachusetts in the US. You get access to Hongene's high quality raw materials and also access to our chemoenzymatic ligation technology. So we develop the process, transfer that to Resi Biopharm, and then the API is assembled in the US. All of the other materials are assembled in the US, And so that's the value that you get. So you get access to Resi Biofarm's state-of-the-art US facilities and top-notch expertise. And they have plenty of experience manufacturing gene editing drugs for clinical programs. So the GMP plasmid, mRNA, LNP formulation, and DP manufacture. That's all bread and butter to them. Just a note on our engineered thermostable T4 ligase. We think this is going to be really important in the future. So the rationale for this is that you can facilitate the reaction at a higher temperature. So if you can do that, then you can reduce the secondary structures, and thereby you can improve the ability of the enzyme to deselect the oligonucleotide fragment impurities that may otherwise be incorporated. So this will help to mitigate some impurities and also incorrectly ligated byproducts. And we are optimistic that we should be able to apply this enzyme to be able to use the C2C strategy, which will help to further drive down the cost, which is going to be so important for these high-volume drugs. So looking at this graph on the right, this is the T4 mutant ligase. This is following incubation at these different temperatures. We're looking at the activity. So this T4 mutant is still active after it's been stored at 52 degrees Celsius. And then if we look at the T4 mutant on the bottom, this is with an actual siRNA with the same type of chemistry, right? It's 2' fluorosome methyls, it's got a GalNAc, and we're looking at the actual activity at these different temperatures, and we see that it's actually maximally active between 52 and 57 degrees, compared to, say, 35 degrees C for the wild-type enzyme. So we think this is going to be a really important lever for us to help achieve our goals. Finally, the controller stereochemistry. So this is something we get asked about a lot, right? Everybody's talking about phosphorothioate stereochemistry and how you're going to control it and stuff like that. And so we're going to do this with HPLC. Obviously, we'll be developing really nice methods there, but we also have a collaboration with Bruker. And through this collaboration, we're taking a phased approach. So the first phase, which is ongoing, we're developing methods for the fragments, for the single strands, the duplex. And then in the next phase, we'll be looking at different synthesis conditions. And then finally, we'll probably end up doing some work to verify our data using stereopure reference materials. And it's very early days yet, but the team at Bruker is making some phenomenal progress. And here's just some examples of the kind of data that they're collecting. And there'll be much more on this to come. But they're collecting 1D, 2D NMRs and phosphorus NMRs. So we were just looking at the batch-to-batch consistency in this particular example. So much more on this to come in the future. Finally, some conclusions. So market demand for siRNA is anticipated to grow really significantly over the next decade and beyond. And this demand, we believe, will be met by ligation chemistry. The advantages are clear: improved quality, yield, environmental sustainability, and scalability. We've manufactured already 1 kilo of this material. It's been in GMP, and it's being dosed in patients now. So it's been past regulators. So we envision actually no significant regulatory hurdles to wider adoption of the technology. Of course, that's a really big statement, and it's really the subject of another entire presentation. C2P and C2C purification strategies, these can be leveraged for large-scale manufacturing to help drive down the costs. And I think that key to that will be technology such as the engineered T4 RNA ligases that will provide those future efficiencies. So oligo fragments, everybody's going to be working on better methods to make these, better technologies. So that's on a work that's in progress. And finally, the high purity single guide for CRISPR-based gene editing drugs, that's accessible by splinted ligation also. And through our partnership with our phenomenal partners, Resi Biofarm. And acknowledgements. So the Hongene oligo team is led by Jim Yang, and the Hongene biocatalysis team is Minju Wei. So I just get to talk about all this stuff. These people obviously do all the hard work, and yeah, my heart goes out to them, and all the respect and best wishes to them. So thank you very much for your attention, and I'll take any questions.
Amazing progress, Dave. Congratulations to Hongene team. So the question is related to the new process which you have, and you said is already in the clinic. So from the specification point of view, you have the new method, which is the ligation, of course. So from a regulatory point of view, did you have to demonstrate that none of the enzymes were actually carried over into the drug substance?
That's a great question. So the CQAs for the ligation process, they're all the same, right? That you might expect for an siRNA manufactured using the Generation 1 technology, with one exception, right? You have the residual enzyme. So we, having been in the mRNA industry, right, we're sort of very well versed in all that kind of stuff, and we've developed a method, really sensitive method with an LOD of 1 ppm, I believe, for detection of that enzyme. It's below that limit of detection, but yes, we did have to demonstrate that, and we have sensitive methods for doing that.
So that's part of CFA?
Yeah.
Thank you.
Yeah. Thanks, Yogesh.
Hi, great talk. I guess my question has two parts. One is I noticed that some of your fragments are quite short. They're like 6 nucleotides long, and you're also incorporating like the 2' fluoros right at the right next to the phosphate for ligation. How do you decide where you're cutting your fragments, and do you find any limitations on what you can put on those sticky ends that you're using for ligation?
Yeah, that's another really good question. So not all of the fragments are created equally, in the sense that there is going to be some combinations that just work better. And we have an algorithm that allows you to sort of do a first pass at that. But we also have, a lot of that is empirical. So we're making lots of different fragments and testing them and trying to figure out which ones work best. But that's a great question.
Thank you.
Sure.
David, that's a great talk. I actually had a similar question like what Yogesh had, but I think the second question I had is about these fragments. Are you considering those as your regulatory starting materials? And are you seeing any off-target effects from those fragments in terms of the efficacy and those things?
Currently, we are treating them as GMP intermediates, but I think in the future there is a really good argument to be made with the regulators for categorizing those as regulatory starting materials, and those are conversations that we'll be having or having in support of our customers. And I know that others in the field in the same field are also having those conversations. As far as off targeting, those fragments are not in the product, right? They're not in the purified product, so there's no concern there. Yeah.
Okay, thank you.
Thanks.
You show great separation in the denaturing method of your product. Have you run that by a non-denaturing method to see if you're getting exactly 1-to-1? Because if you miss one of the sides, you're going to get maybe one or the other. And I know I've seen excess scent strands in some of these, and I was wondering if you see anything, or if you've done this type of analysis.
It's a great question. I was thinking about that last night. I was like, I should have been putting the slides on the non-denaturing methods. So we have a non-denaturing SEC method for the divalent siRNA, and a non-denaturing reverse phase method for the inclisiran example. So yeah, they are very clean. We've spiked in single strand. For the divalent siRNA, it's very, very, very interesting actually. We see like a partial duplex for that. So we see a little shoulder for that one. We just figured out what that is. But yeah, the non-denaturing chromatography does look very good as well. You have to trust me about that.
Oh, no, I believe you, of course. Thank you.
Thank you.
Hi, thanks for the talk. For the guide RNA, did you consider or have a look at whether you could use the areas of duplex that are already formed by the guide rather than the splints?
We did take a look at that, but we also, the splinted ligation just works so well. It just works spectacularly well. And you know, the DNA splints are really simple for us to make, and simple to put in the reaction, simple to digest, and we get better quality with that process than we would from the process, using the secondary structure, the natural secondary structure of the single guide. But it is something, something we've looked at, something we will continue to look at as well, because there could be advantages, right, in terms of the process for that. You're not using splints, and that could be an advantage, yeah.
Thanks.
Thank you. That's it. Well, if that's it, thank you very much. Appreciate your attention.